Hello everyone, my name is Yulia Yakovleva. I present our work named Black Cat in a Dark Room, Search for New Viruses in Metagenomes. Detection of hidden viral diversity is a challenging task which goes beyond the standard protocol of processing metagenomic data. Meanwhile, publicly available databases contain a large amount of metagenomic data, which remains largely understudied. Previous works suggest that we underestimate the diversity of viruses on our planet. The figure on the slide represents accumulation curves showing the protein cluster growth with increased sampling. Green, blue, and red represent clusters from all aquatic and human metagenomes respectively. The curves of cluster accumulation in the two most heavy sampled habitats, human-associated and marine, appears to reach saturation. However, the rate of cluster discovery doesn't plateau when all samples are considered, suggesting that the global viral sequence space is largely uncharacterized. Thus, the goal of our work is to design and to test the new pipeline for detecting full-length viral genomes from assembled metagenomes. Significant part of viral genomes represent cyclic or linear molecules with the ends containing repeated sequences. Both types could be recognized as cyclic sequences. We detect such contexts by searching repeats, ranging from 50 base pairs by Knuth-Morris-Pratt algorithm. This approach takes linear time depending on the maximum length of the allowed repeat, which permits to process large amounts of data and reduce its dimensionality. We classify cyclic sequences as viral or non-viral based on the predicted gene content using the viral verified tool. For each selected viral contig, we identify the capsid and terminase genes based on HMM profiles. We aligned found protein sequences against another NCBI database with Diamond. The protein sequences, both queries and hits, belonging to each HMM profile, were clustered with CD hit. On the resulting centroids, phylogenetic analysis was performed. The completeness of viral contigs was inspected with viral complete, followed by an annotation. A large number of proteins and a high level of similarity among some sequences requires the clusterization step to reduce the number of data for phylogenetic analysis. For this reason, we used CD hit with cutoff span 80% and identity 50%. We divided resulting clusters on three groups: clusters which contain only database hits, marked blue; clusters which contain predicted proteins without database hits, marked green; and mixed clusters which contain both database hits and predicted proteins. Analysis of available metagenomes from NCBI database reveals that the largest part of metagenomes belongs to human. These metagenomes are known to be quite well studied, so we decided to focus on marine, seawater, soil, and biofilm habitats. During the analysis, we were faced with an incredible amount of data, more than 170 gigabytes, which takes a lot of processing time in case of using existing approaches. We found hundreds of structural viral genes from different protein groups. The most abundant were terminase genes, both for marine seawater and soil habitats. We received protein alignments for each HMM profile. Large alignments containing centroids were analyzed with UPGMA method and smaller were analyzed with Raximate tool. The slide demonstrates three based on analysis of P22 code protein. Notably, some clusters contain hits of bacterial origin. We assume that this is not an artifact because viral verified discard contigs, which could be a plasmid or chromosomal origin. Moreover, we propose that hits taxonomy could refine the taxonomy of potential virus host. These slides demonstrate three based on the analysis of terminase 6 genes. Green color indicates protein clusters which form separate groups. These clusters are of particular interest because they might belong to putatively new groups of viruses. We found 879 and 325 viral contigs for marine seawater and soil habitats respectively. The length distribution of these contigs before and after assessing genome completeness are shown on the slide. This check helps us to eliminate false positive contigs with small lengths, presented in Figure 1. The distribution shape of received viral contigs, presented in Figure 2, is similar to shape of distribution DNA viral genomes from RefSeq database, presented in Figure 3. Notably, the distribution is multimodal. Most of the founded viral contigs belong to phage genomes. Since a large number of results could not be thoroughly inspected manually, we used biofilm metagenomes as a small dataset. A biofilm is the thick layer of prokaryotic organisms that have aggregated to form a colony and are immersed into the extracellular matrix. Biofilms may form on living or non-living surfaces and can be prevalent in natural, industrial, and hospital settings. Up to date, there are 2 biofilm metagenomes in NCBI assembly database. Our pipeline detected 272 circular contigs in these metagenomes. 5 of them were classified as putatively new viruses. We discarded the 2 contigs from the following analysis as they were too short. The next 3 slides demonstrates maximum likelihood trees based on alignments of amino acid sequences of terminase proteins of putatively new viruses identified in this study on the right and genome annotation on the left. A newly detected virus, marked by orange point from untreated reverse osmosis biofluid membrane, forms a distinct branch in the Cipabrida tree. The next virus, extracted from the same setting, demonstrates discordant phylogenetic signal depending on analyzed genes, terminase at the top and major capsid protein at the bottom. While terminase sequence shows similarity with the protein of already known bacteriophage, major capsid protein sequence forms a separate branch. Such mosaicism in virus genomes makes it difficult to determine the position within a new or previously described group. And the last virus, extracted from the vanillin-treated reverse osmosis membrane biofilm, belongs to Podavirida family of viruses. To conclude, our approach allowed us to detect full-length viral genomes with a lower chance of false positive results. In the future, the user of our pipeline can submit metagenome assemblies or raw reads to the input and receive annotated viral genomes from the data. Thank you for your attention.